Hello, everybody. It's great to be here. I want to thank the organizers for inviting me. This has just been a phenomenal meeting, and I'm going to wrap up the microbiome session. Like Mike said, I'm from the Jackson Laboratories in Connecticut, not Bar Harbor. We have a brand new campus in Connecticut that's devoted to human and medical research. So we've heard that this is sort of an era of microbiome research. At the upper left is another one of these pictures of publications, which is in exponential growth phase, and that really took off right around the time that next generation sequencing came along, because as you heard earlier, the key to microbiome research is to take a complex mixture of organisms and not culture them, just sequence the heck out of it and figure out what's there. Couldn't do that very well with Sanger sequencing. That's also when the Human Microbiome Project of the NIH started, which I'll tell you about in a little bit. The upper right are funding from the NIH, which you see is also in growth mode, so that's a good thing. And then down at the bottom, just a couple of weeks ago, the National Microbiome Initiative was announced by the White House, which is another $100 million or so from government agencies that'll go into microbiome research. And they also, the White House also did a survey and came up with at least $400 million in other microbiome initiatives in academia and industry that are going on right now. So we're definitely right in a golden area for microbiology and for microbiome research. Another thing, we've sort of talked about bacteria in the talk so far today, but it's not just bacteria that is part of the microbiome. There's viruses, and the histogram there is the average number of viruses, viral genera, that we find in a healthy person looking at their stool. So all of you have not only 10 times more bacteria in you than you have human cells, you're also infected. So, but you're happy. And what are the viruses doing is one of the big questions for microbiome research. There is a New York Times article from today. That's our favorite science journal in the microbiome research area. Could Alzheimer's stem from infections? It makes sense, experts say. So there's a lot of interesting thinking about viruses, inflammation, and diseases, particularly neuromuscular, other kinds of diseases like that might end up from chronic infections. And then down at the bottom, Parkinson's disease and alpha-synuclein is Parkinson's disease a prion-like disorder? So even beyond organisms is the idea that somehow the microbiome might be creating misfolded proteins, and over decades and things like that, something happened. Parkinson's disease can be preceded by constipation for decades ahead of time, and suggesting that there may be something in the gut that's contributing to it too. So this is a wide open field. You can just connect just about anything potentially to the microbiome at some point, and that's what the challenge of the research is. Now, I had the honor of working on both the Human Genome Project as well as the Human Microbiome Project, and since all the stuff that came before this session really was focusing on the human genome, it's useful to have some thoughts about how these two areas compare, because they're basically genomics. One is of your microbes and one is of your genome. The human genome, of course, is locked into a sequence pretty much, except for somatic mutation, so it's not variable, whereas your microbiome can be variable. Diet, all kinds of other things can change the organisms you have, so that makes it a little challenging. You have the same genome in all your cells, whereas in different parts of your body, you have different microbiomes because the conditions are different. Aerobic, anaerobic, dry, moist, nutrient-rich, nutrient-poor, oily, things like that. So that's another challenge. On the good side is the fact that gene therapy and things like that, where you'd like to intervene with the human genome, is tough. But we can do microbiome transplants, we can manipulate the microorganisms, and so the possibility for doing therapeutics through the microbiome is very real and very close to us right now. In fact, there are some standard of care for treating C. difficile-associated diarrhea is the famous fecal microbiota transplant. So let me tell you a little bit about the Human Microbiome Project. This is Human Microbiome Project 1, because there's a 2 that's going on right now as well. This was 300 subjects that were recruited. They all came in for 2 or 3 samplings, so we had a total of 700 sampling visits. The boys were sampled at 15 different body sites. The girls were sampled at 18 different body sites, and this led to close to 12,000 samples that we collected to analyze the microbiome. And then there was an extensive set of metadata that was collected about each of the subjects as well. And all of this is available through dbGaP and NCBI. So then there's 2 types of sequencing that you do in microbiome research. You sequence the 16S ribosomal RNA gene, which is how you define a bacterial species. It's 16S sequence. So when you do that, you're taking a census of the bacteria that are present. And we typically sequence 7,000 reads per sample, ended up with 81 million reads, 40 gigabases or something like that. Not super big data, but you can kind of think of that as like whole exon sequencing or something like that. Metagenomic shotgun sequencing is the powerful sequencing where you sequence everything that's there. And there we went for 100 million reads for each sample. We ended up with 45 terabases of data, and that you analyze for everything: bacteria, viruses, fungi, you name it. So, just some thoughts about this. Most of the studies in microbiomes have tens of subjects because it's hard to recruit people with different diseases, and there aren't many really huge studies the way there are with human genomic work. Of the 700 sampling visits, we had an extensive exclusion criteria so that these were all healthy people. We gave them a little kit of soap and deodorant and stuff, like toothpaste to use, so that we tried to make it as uniform as possible. But you can't control diet, you can't control genotype, and so there's a lot of inherent confounding variables that were in that. And then, as was mentioned, we tried to keep sequencing and data production consistent between the centers that were involved, but sequencing technology changes. There are degenerate primers, so slight differences in PCR procedures affected. So there's all kinds of biases creeping into this, but this was about the best that we could do. And despite all of those things, it works pretty well. This is another one. This is the lung human microbiome project, where we look at people who have an HIV infection and have a bloom of their microbiome in their lung and then get antiretroviral therapy. The virus goes away, but the bloom takes a while to get back to normal, and in the meantime they can develop COPD or other consequences. So if you look at these dots, each dot is a community from a different individual. They start out, when they have HIV, they're green, they're spread out through the PCOA plot, and over several years, they all come very similar to each other, back to sort of a normal or a post-HAART microbiome. So we can see things that make sense when we look at microbiomes. Here's another one where we have a model for C. difficile infection in a mouse, and a fecal transplant makes a sensitive mouse into a resistant mouse. That's what the sensitive mouse is the red line. The resistant after the transplant is the black line at the top. And microbiome analysis allowed us to zero in on 4 particular bacteria that when we just give just those 4 bacteria, we can get almost as much protection. So, we can learn things, and there's hope for developing therapeutics and things like that, although mouse C. difficile in this particular model is probably quite different than human. Okay, so how to get this into the clinic, how to have an impact on our topic, our favorite topic for this meeting. So there's a whole ecosystem of analyses that we do on these data sets to look at 16S, to look at shotgun, to look at biochemical pathways, different types of microbes. All that stuff is going on. But we also have the host, and we have to focus on the host because the microbes are not doing what they're doing by themselves. There's a dialogue going on. So Mike Snyder and I have an HMP2 project to look at pre-diabetics who convert to becoming diabetes. We have 100 subjects in that. They're analyzed longitudinally for 3 years. There's all the microbiome stuff, but there's also many, many host side things that are being measured. And the challenge here is integrated analysis. In fact, the challenge is really how to put this stuff in databases that can be used in a sensible way for analysis too, because all the data types are quite different. That's certainly the future. And then finally, if we think about what the whole picture looks like, the stuff we're talking about are the things in the gray box there, getting samples, doing the sequencing and the analysis of that, finding organisms of interest. But then there's all the other things. What about the metabolites from those organisms? I showed you one example of making a mixture of organisms that had some bioactivity. And how do you test all of this is the big challenge. How do you prove causation? We're really right up against now trying to figure out how to do more human studies. The mouse is the favored model, whether it's a germ-free mouse or not a germ-free mouse. I showed you for the C. difficile model how you could use that. What's the relation of that to what's really going on in a human? Obviously, there's big differences, but to learn any mechanisms of how the microbiome interacts with the host, even if it's more slanted towards a rodent physiology than a human physiology, is huge right now in terms of being able to figure out how to look at it in humans. So I will stop there. Thank you.